Okay, coming. Voice is coming. Check it out. Hello. Is my voice audible? Come on, come on, students. Okay, done. See, around the cell there should be a plasma membrane. Then second one is cell wall. Around the cell wall, some bacteria possess some extraordinary structure or some kind of extension. That extension is known as glycocalyx. In this, three layers of envelope has been developed around bacterial cell. These three layers of envelope, one by one if you check. First one, plasma membrane. Second one, cell wall. And the third one is known as glycocalyx. The three layers. These three layers collectively known as cell envelope. Three layers are collectively known as cell envelope. Let us talk about them one by one. First one, what is the story of glycocalyx? Glyco means carbohydrate, calyx means outer covering or rigid covering. That's called glycocalyx. Glycocalyx is a kind of polysaccharide coating. Polysaccharide coat around some bacteria. Not all bacteria possess glycocalyx. Most of the bacteria without glycocalyx they can survive. If a bacteria possesses glycocalyx, it is made up of polysaccharide. Next one. Glycocalyx will be on the basis of its physical composition. Sometimes it is flexible. Sometimes it is tough and rigid. Very rigid and tough. If it is a flexible layer, the glycocalyx is having a name called slime layer. Next, if it is a tough and rigid, that's called capsule. Listen, these things we have come across in molecular basis of inheritance. If you recall it very closely, when we were talking about molecular biology, transformation principle that was discovered by Griffith we had discussed. There, S strain, R strain type of streptococcal pneumoniae we have mentioned. There, S strain is virulent, R strain is non-virulent. What is this S strain and R strain? Those which are having the glycocalyx are called S strain. Those which are not having glycocalyx are called R strain. Simply to remember, in coordination to that information, generally those bacteria which are having glycocalyx are virulent bacteria. Many bacteria which are having the glycocalyx are capsule or slime layer. They are virulent in nature. Glycocalyx bacteria virulent or pathogenic in nature. Pathogenic. This one is there in our NCERT book, that's why I am saying, but it doesn't mean that every bacterium which is having glycocalyx definitely would behave like a pathogenic. But generally pathogenic bacteria are having glycocalyx coat on the top of them. Okay? Then come back to next one, cell wall. Cell wall. First question, do all bacteria will have cell wall? No. There is a group of bacteria known as mycoplasma. Mycoplasma is a bacteria, or it is the bacteria without cell wall. They don't have cell wall. Which one? Mycoplasma. Very good. If they don't have cell wall, how could they survive? Be careful. In order to survive, it is not compulsory to have cell wall. Cell wall is there, it gives some advantages. It doesn't mean that a bacteria will survive only by the presence of cell wall. That's not the thing. Then come back. Cell wall, if present, made up of what? Made up of peptidoglycan. Peptidoglycan, commonly known as murine. Murine or peptidoglycan. Next one, it is a rigid layer. First thing, shape of cells, shape of cell is due to cell wall. Shape. We have seen bacteria possess variety of shapes like bacillus, rod shape, cocci, spherical, vibrio, comma shape. Variety of shapes will be there. All those shapes are developed due to presence of cell wall. Cell wall is the reason behind the shape of the cells. If so, earlier you said something called mycoplasma and you have denoted mycoplasma do not have cell wall. Does it mean that the mycoplasma will not have any shape? Of course. Okay? Mycoplasma pleomorphic in nature. Pleomorphic in nature. Pleomorphic in the sense they don't have proper shape. The bacteria that does not, or a cell that does not possess any proper or particular shape, that bacteria is known as pleomorphic bacteria. Next one. It is also, cell wall protects the cell from bursting and collapsing. Bursting and collapsing. What does it mean by this bursting and collapsing? Imagine I have taken a cell kept in hypertonic solution. That means around the cell, the concentrated solution is present. Whatever the water present in the cell started to leak out. As a result, cell shrink. That shrinking process is known as collapsing. In addition to that, if you keep a cell under hypotonic solution, that means water rich solution, water started to penetrate inside the cell. As a result, there will be a kind of swelling will happen inside the cell, that's called bursting, or eventually cell is going to burst. Like this, cell neither burst nor collapse immediately due to change in external environmental solutions. That would be the protection given by cell wall. Then go to most important part, that's called plasma membrane. Now one thing about that, whenever we see plasma membrane is the universal structure for a cell. In fact, without plasma membrane, there is no cell. So, do all cells have similar type of plasma membrane? Almost same. With some minor additions and subtractions will be there in the nature of chemicals present in the plasma membrane, but almost all kinds of cells will have same structural orientation in the plasma membrane that indicate structurally similar to eukaryotic cell plasma membrane. That's why whatever the details that we will study about plasma membrane, we will study about one time about plasma membrane. Okay, done. Here, after cell envelope primary discussion, we should talk about one matter that's called Gram staining. Gram staining. There was a scientist called Christian Gram. he added a chemical or a chemical stain on the top of bacteria called crystal violet. Crystal violet, commonly known as gram stain. He had taken few drops of crystal violet, added on the top of bacteria. Some bacteria had absorbed this color. Some bacteria couldn't be absorb the color. The bacteria which absorbed color, so-called crystal violet color, he named them as gram positive bacteria. Some bacteria couldn't absorb the color and named it as gram negative. Simply, if crystal violet absorb gram positive, they intake or absorb the color. They can intake or absorb color. Gram negative bacteria cannot absorb color. That altogether makes gram positive bacteria will have thick cell wall. The cell wall is very thick. Gram negative bacteria is having the cell wall that is thin. Cell wall very thick in gram positive, very thin in gram negative bacteria. This might be clearly understood like this. When we add few drops of crystal violet, those bacteria which are having thick cell wall can absorb that color, retain the color for future purpose, or else in future the color will be present in the cell wall. But some cases, immediately after absorbing the color, a cell wall could not retain that color for future. This we will call it as gram negative. The earlier one is called gram positive. Altogether, thick cell wall containing bacteria can retain the color, called gram positive. Thin cell wall containing one cannot absorb the color. This is gram staining procedure. I did not explain the procedure of gram staining in completely, but this much information fine to understand the information that they have given in our NCERT book. That much fine. Then go back to cell. On the surface, what are the structures we will see? On the surface of cell envelope, we will see structures like flagella. Next one, we will see some structures like pili. These are pili, and some more structures like fimbriae. The way I am representing these structures on the top of the bacteria, it is essential. 3 extensions will be there on the top of the envelope. One is flagella, pili, fimbriae. What are those extensions and their usage? First one, I will talk about pili. If you take pili, which is also known as sex pili. Why? This is a kind of tube or tubular structure, help in conjugation process. Simply to understand, in bacteria, there are some modes of sexual reproduction. Exact mode of sexual reproduction may not be identified in bacterial cells like meiosis, fertilization, zygote formation. These events are not there. But prokaryotic cells do have their own mode of sexual reproduction in order to develop variations. The sexual mode of reproductions are conjugation, transduction, transformation types. In that, conjugation is a kind of physical association of two bacteria. Two bacteria will come close to one another and they will join by formation of a conjugation tube. That conjugation tube is formed by pili. Since it is helping for sexual reproduction in bacteria, that's why sometimes it is called as sex pili. Very important. Apart from that, Pili has no other role to be conduct. Next one more is there, Fimbriae. Fimbriae are bristle-like structure. Bristles. Bristles means small hair-like structures. What is the bristle-like Fimbriae work? They help in attachment of bacteria on solid surface or on substratum. Generally, this Fimbriae will be present on those parasitic bacteria. In order to make them to attach, the bacteria has to attach on the surface of host cell. To make itself cling or attach it to the host cell, the bacterial cells develop some special kind of bristle-like structure, that is fine hair-like structures. These hair-like structures are known as Fimbriae. Apart from that, Fimbriae also do not have any work. Next one, Pili made up of a protein called pilin. Both are protein structures. Protein, pilin. Fimbriae made up of fimbrin. Either you take Pili or Fimbriae, they are made up of proteins. But one interesting common thing about them, both do not work in locomotion. Not useful for locomotion. Locomotion of bacteria is not possible due to Pili or Fimbriae. Now, how does a bacteria can be motile? Careful. In a eukaryotic cell, there may be so many varieties of locomotory organs are there. In fact, eukaryotes will have flagella, cilia, and in rare cases, pseudopodia may also help in locomotion. But a prokaryotic cell will have only one kind of locomotory organ, that's called flagella. Flagella. Flagella plural, flagellum singular. Flagella present in locomotory or those bacteria which are able to be motile. All will have one thing, that's called flagella, nothing else. Imagine, there will be some bacteria who cannot motile, there will be some bacteria who are motile. All motile bacteria possess flagellum. Flagellum is the one which is exclusively belongs to motile bacteria. You have seen a bacteria with flagella. That bacteria definitely can able to show motility on its own. Next one, the flagella made up of flagellin protein. It is also made up of flagellin protein, which means 9+2 microtubule arrangement absent. 9+2 array of microtubules is absent. Why do you say like that? Simple. When we talk about eukaryotic cell, a eukaryotic cell flagella made up of microtubules. Microtubules are exclusive characters of eukaryotic cell. In that case, in case of prokaryotic flagella, motility and the characterization may be same, but internal structural orientation is completely different. Prokaryotic cells will have flagella simply made up of flagellin protein, but in eukaryotic flagella, microtubules are present which are arranged in the form of 9+2 arrangement or 9+2 array of microtubules will be present. Keep it in mind, Later, when we will talk about end of the story in the cell, the unit of life, we will have flagella structure in eukaryotes and also centrosome structure. Those structures we will in detail discuss. But altogether, whenever we talk about prokaryotic flagella is made up of flagellin protein, but that protein never follows any one particular mode of arrangement of microtubule like structures. They just protein filled large filament will be present. In this case, prokaryotic flagella will have three structures. Flagella possess three structures. One is basal body, that is the where flagella originated. Next one, hook, very good. Next one followed by filament. In this case, first one, if you talk the basal body, where does it present? The basal body present in plasma membrane. Within the plasma membrane, there will be a presence of basal body. This is the basal body, for example. Actually, from plasma membrane, basal body originated. Gradually, it will cross our cell wall, and also if glycocalyx present, glycocalyx also. But overall, the basal body will comes out of cell wall. That's why always this flagella is started to appear, it is coming from cell wall, but actually flagella will come from plasma membrane. Origin is plasma membrane, but externally if you see, the flagella started to appear like it is comes out of cell wall, because if you see from the outer surface, first you will see cell wall, then it appears like something is protruding away from cell wall. That protruding body we can call it as flagella. But actually flagella would be originated from plasma membrane, because the basal body will be present at plasma membrane only. Cell wall, something dead, which could not be generate anything. Cell wall itself is secreted by our plasma membrane. So, if something is growing on the top of the cell, that material must be secreted by our plasma membrane. So, on the cell wall, there is a presence of basal body. That basal body will also having hook, then followed by filament. This is the filament. This one is hook, and this one is basal body. These are the parts. Next thing, do all bacteria have flagella? No. What type of bacteria will have flagella? Simply, whatever the bacteria which can able to motile, that bacteria should be having flagella. Flagella has no other work at all. They have only one work. That's called locomotion. Keep it in mind. But another thing, location and number of flagella varies from cell to cell. Location as well as number of flagella varies from cell to cell. Some bacteria might be having single flagella at one end. Some bacteria may have two flagella at the opposite end. Some bacteria might be having a bunch of flagella at one end. Some bacteria will have flagella all over their body. Like that, number distribution may vary from cell to cell, but in all, there will be a variations in the number of flagella and also the location of flagella present inside a bacterial cell. That's about the flagellar arrangement. Then, after the flagellar structure, let us see what they are talking other structures. In internally areas, you will see the plasma membrane of bacteria will started to enfold. Plasma membrane started to enfold. These enfoldings of the plasma membrane are known as mesosomes. Mesosomes. Let us learn some details about mesosomes and their importance. First one, who are mesosomes? Mesosomes are unique to prokaryotic cell. I'm not saying all prokaryotes will have mesosomes. That's absolutely wrong. Some of the prokaryotic cells do not have mesosomes. But if mesosomes present, what is their orientation and their functions? See, mesosomes are enfoldings of plasma membrane. Mesosomes are enfoldings of plasma membrane. What do you mean by enfoldings? Plasma membrane itself will fold it towards inner side, and it is making something is called mesosomal structure. In that case, shape will be tubular sometimes, tube-like structures like this, tube-like structure. Sometimes they are vesicular structure like this. Sometimes even they are lamellae. Lamellae means membranous sacs called lamellae. Like that, shape of mesosomes may vary from cell to cell. They don't have a constant shape because they are enfoldings of plasma membrane. Then important thing to be noticed there, their functions. Functions of plasma mesosomes. First and foremost function, cell wall formation. If you recall, cell wall of the bacteria made up of peptidoglycan or murine. That murine or peptidoglycan chemical synthesized by plasma membrane, and it would be secreted outside. That's why cell wall formation is under the control of our mesosomes. Next one. Now compare this one. If you could recall, what was the cell organelle in a eukaryotic cell, especially plant cell, responsible for cell wall formation? That was Golgi complex. Golgi complex in eukaryotic cell will help in cell wall formation, and in case of prokaryotic cell, mesosomes responsible for the same cell wall composition. Next one. They also help in cellular respiration. Cellular respiration. Don't mistake respiration means exchange of gases. No. Exchange of gases means breathing. What about respiration? Respiration is a technique inside the cell where food get oxidized in order to release energy in the form of ATP. That is called as cellular respiration. Okay. Respiration require gases. That's why multicellular organisms can have some separate variety of respiratory organs only for the sake of breathing. Come back to the story. If you are talking about mesosomes at the site of cellular respiration, what does it mean by that? They are responsible for synthesis of ATP. Synthesis of ATP. When it comes to the word ATP, you can directly correlate in a eukaryotic cell, the ATP machinery is mitochondria. Obviously, more mitochondrial function, more ATP can be synthesized by a eukaryotic cell. Now the question under our level, does the prokaryotic cell require ATP? Of course. No living cell can survive without the requirement of ATP. If mitochondria is absent in prokaryotic cell, there should be someone to be compensating that loss. That someone is nothing but mesosomes. If you closely observe mitochondrial function, cellular respiratory events like link reaction, Krebs cycle, and also electron transport chain. Link reaction, Krebs cycle in the matrix, link reaction, Krebs cycle in the matrix, electron transport chain might be happening on inner membrane. That inner membrane electron transport chain, do you think is it possible in prokaryotic cell because they don't have mitochondria? Of course possible. That would be happen on membrane of mesosomes. Be very careful. Almost all activities which are conducted by a eukaryotic cell, a prokaryotic cell can also do. But the different places in the different locations, it is possible in case of prokaryotic cell. If anybody asks you a question, does a prokaryotic cell can conduct electron transport chain as like the mitochondrial inner membrane? Of course, mitochondria is absent, but mesosomes are compensating the work of mitochondria in a prokaryotic cell. Very good. Next one. Another work of mesosome, that is DNA replication. DNA replication. Now, I wanted to say one thing about DNA replication. Recall how much time a typical bacteria takes for the cell division or binary fission. Simply, it will take around 20 minutes only. In that 20 minutes, bacteria spend 18 minutes only for the DNA replication. If you could remember, I said bacteria possess circular double-stranded DNA. That circular double-stranded DNA is present in the form of nucleoid or naked DNA. That naked DNA is not always freely present in the cytoplasm. Mostly it is associated with mesosomes. Why it has to be associated? The quick way of DNA replication is possible during at the time of cell division or binary fission with the help of mesosomes. Whatever the enzymes required for the DNA replication, all the enzymes would be provided by our mesosomes. As a result, bacterial DNA can be replicated. Mesosome function not only ends to support DNA replication, even after the DNA replication, the mesosomes will support the replicated DNA equally distributed to two daughter cells. What does it mean by that? Let us see one thing. Distribution of replicated DNA to daughter cells. Why are you saying this? This is very important. Many people I have seen constantly misunderstanding. What does it mean by distribution of DNA equally to two daughter cells? Let us see. In a eukaryotic cell, how does distribution occur? If you consider a eukaryotic cell, in a metaphase, there is a clear-cut development of two chromatids in one chromosome. These two chromatids, each chromatid contain one DNA. That DNA has to be entered in future daughter cells. There will be a phenomena called spindle fiber attachment. On each centromere, both sides, you know these things, that's why I am simply moving. There is a centromere. Both sides of the centromere, one side here one kinetochore, another side another kinetochore will be present. For both kinetochores, opposite pole spindle fibers will come and they will attach. During the anaphase, they will drag these two chromatids away from each other. As a result, centromere split, chromatids separate, and each chromatid will move to opposite pole. As a result, the distribution of DNA that was replicated during S phase possible in case of eukaryotic cell. This much long story is not possible in prokaryotic cells. Why? There is no concept of spindle fiber formation. If you recall, the prokaryote cells undergo amitosis, which means there will be no formation of mitotic threads. Mitotic threads are absent in case of prokaryotic cell. So there should be some way we can give that replicated DNA molecules equally to two daughter cells. That way is through mesosomes. Mesosomes are the one who will split the replicated DNA and given to two daughter cells equally. This is the reason why mesosomes help in distribution of replicated DNA equally to two daughter cells. Am I clear the word? Next one. One more function, cell secretion. What does it mean by cell secretion? Anything that should be goes out of the cell. In case of eukaryotic cell, the secretion is under the control of a special cell organelle called Golgi complex. Later we will talk about it. With respect to Golgi complex, wherever a cell secretes something away from the cell, anything that goes out of the cell, that is strictly regulated by Golgi complex. But in prokaryotic cell, Golgi complex is not there. So somebody should be there to compensate that work. That work is done by our mesosomes also. Okay, I mean mesosomes do that work also. So observe the statement here: cell wall formation, cellular respiration, DNA replication, replicated DNA distributed to daughter cells, and also cell secretion. All these activities are under the control of mesosomes. Mesosomes, if not present, there may be different types of functions conducted by different places, but mesosomes, if present, they are the one who do all these activities. Next one. Apart from the mesosomes, some other things we need to talk about. A prokaryotic cell membrane. On the membrane, here and there, we will see some parts which possess pigmented area, pigments. Okay, those pigmented areas in plasma membrane are known as chromatophores. They have a special name called chromatophores. Who are these chromatophores and what is their story? Chromo means color. Let me take the chromatophore story for us. They are present on membrane. Be careful. Chromatophores are not freely present in cytoplasm. Chromatophores present on membrane. They are membranous areas. In that case, what kind of structures do you call as chromatophores? Chromatophores, pigmented membranous structures. Pigmented membranous structures. They are pigmented membranous structures. What does it mean by pigmented membranous structures? On the top of the plasma membrane, here and there, some amount of photosynthetic pigment will be deposited. Here very interestingly you have to remember, those pigments that we will see in chloroplast will not be present in chromatophores. Typically, a eukaryotic chloroplast consists of that chlorophyll A, B, and also carotenoids, xanthophylls. But chromatophores do not have all of them. They have special, that is chlorophyll. Here also rarely chlorophyll A present in case of blue-green algae. Remaining bacteria will have bacterial chlorophyll. It's a special one. I will discuss about the bacterial chlorophyll and chlorophyll A differences when we begin the photosynthetic machinery, but keep it in mind. Normally bacterial chlorophyll will present on chromatophores. In addition to that, there is another kind of pigment called phycobilins or some accessory pigments. Phycobilins will be there in cyanobacteria. Phycobilins in cyanobacteria, blue-green algae, we would see them. Blue-green algae are prokaryotic bacteria. Okay, in that, except this, you don't see carotenoids, xanthophylls. Don't construe or mistake by just listening. Chromatophores contain photosynthetic pigments, not normal photosynthetic pigments as like our plant cells possess. Plant cells will have a different composition of photosynthetic pigment, and bacteria or prokaryotic cell will have different composition of photosynthetic pigment. That's why I am emphasizing that. Anyhow, if a eukaryotic cell performs photosynthesis, that is due to chloroplast. If a prokaryotic cell performs photosynthesis, that is due to chromatophores, because a prokaryotic cell does not possess chloroplast. Still they can maintain photosynthetic machinery. Next one. Inside a prokaryotic cell cytoplasm, we would see some bodies called inclusion bodies. Inclusion bodies. Who are this inclusion? Why did you say they are inclusion bodies? Inclusion bodies means they are freely present in cytoplasm in a disappeared state. That means how many present, where they are present, you cannot significantly notice them. That's why they are calling them as inclusion bodies. Okay, done. What is their main work? These are non-membranous storage granules. Non-membranous storage granules. They just meant for storage. It doesn't mean that what they will store only particularly. Anything that will be stored inside the cytoplasm of prokaryotic cell generally noticed as inclusion bodies. My dears, remember, I have seen one statement in NCERT book, you could also see. Something is unique present in the form of inclusion bodies in prokaryotes. Be careful. If they ask, prokaryote cells only will have inclusion bodies, that is absolutely wrong. Why they say prokaryote cells will have something unique in the form of inclusion bodies? There are inclusion bodies in prokaryotic cell. Such kind of inclusion bodies we do not see in eukaryotic cell. That is the intention. Other than that, inclusion bodies are not unique to prokaryotic cell. You would see this statement in NCERT book like this. Something unique present in prokaryotic cell in the name of inclusion bodies. Something unique. That means this thing slightly different from that of eukaryotic cell. If you come across a statement like inclusion bodies exclusively present in prokaryotic cell, absolutely wrong. Why? Inclusion body is not something that is any cell organelle. Anything that it is included or anything that is inclusively present in cytoplasm, you can have inclusion body title. Okay, so come back to the inclusion bodies that we would see in prokaryotic cell. Over here, there are some phosphate granules. Some bacteria store phosphate. Some bacteria store sulfur granules. Some bacteria store iron granules. In addition to them, there is a presence of glycogen granules. This is very important. Glycogen granules. Glycogen, what does it mean by this glycogen granules? I think a fundamental statement we know, plants store food in the form of starch. Animals store food in the form of glycogen. This we are studying from 8th class or 7th class onwards. Now here the question will arise, other than animals, is there any member store glycogen? We don't have just only animals in this world. In addition to animal, we have fungi, we have bacteria, we have protozoans, protista, variety are there. So, in addition to animals, is there anyone who store glycogen? Okay, keep it in that question in the mind. Next one. In addition to plant, is there anyone store starch? If you closely analyze these questions, generally, other than plant, remaining store glycogen. Glycogen is a commonly storage food material. Glycogen stored by bacteria, glycogen stored by fungi, glycogen stored by protista member. Wherever you come across a body that it is trying to store food, especially in the form of carbohydrate, that food will be stored in the form of glycogen. But when it comes to plants, plants are different things. Plants store starch because starch would be stored in dry form. Water deficit form, carbohydrates can be stored. That water deficit form is known as starch. Water required for other kind of carbohydrates to store, that water required components are called glycogen. In our body, we will always store glycogen. Bacteria store glycogen. Fungi store glycogen. Protista store glycogen. But plants store starch. That's why they have emphasized in our lower class books, plant store starch, plant store starch. Why? Because plants only store starch. Remaining all are storing glycogen. Next one. Bacteria also store glycogen. In addition to this, there is some other information we need to learn. Gas vacuoles. Who are these gas vacuoles and what are their story? Observe. When you talk about the term vacuole, did you see the vacuole term only in our bacteria? No, earlier we have seen there is a name called vacuolar membrane which is called tonoplast. Inside the tonoplast there is a place called tonoplasm and contractile vacuole, and also there is a vacuole called, in case of amoeba you have to remember, that is contractile vacuole and also food vacuole. If amoeba perform or any other protozoan has performed phagocytosis, that food stored in the form of vacuole, that vacuole is known as food vacuole. And some protista members will have contractile vacuoles. The contractile vacuoles will help in osmoregulation, water maintenance. Getting the idea? So altogether, wherever you come across, generally vacuole, don't include that vacuole, gas vacuole. Vacuoles you should remember 3. Sap vacuole, that's tonoplast, sonoplasm concept. Next one, contractile vacuole, help in osmoregulation. One more vacuole is known as, food vacuole. Over there, you should never include something that is called gas vacuole. Why? Gas vacuoles are air bubbles covered by protein rich membrane. They don't have membrane in fact, or protein rich layer. That's called gas vacuoles. Gas vacuoles unique to prokaryotic cell. You will never find gas vacuoles in any kind of eukaryotic cell. Somewhere else if you find gas vacuoles, be careful. That organism is nothing but a prokaryotic cell, because in a eukaryotic cell you will find three types of vacuoles: sap vacuole, food vacuole, contractile vacuole. Nothing else in eukaryotic cell. Now, prokaryotic cells will have gas vacuole. They are unique to prokaryotic cell. Not all prokaryotic cell, only in photosynthetic prokaryotic cell. Not all prokaryotic cell, only in photosynthetic prokaryotic cell. What is the relation between the photosynthesis and also the presence of gas vacuole? You are saying here photosynthetic prokaryotic cells are having gas vacuole. Extraordinary. Okay, we believe. What is the relation between a gas vacuole and a photosynthetic bacteria? Simple. In order to constantly expose to the sunlight, some bacteria stay on the surface of the water in aquatic environment. In order to keep themselves on the surface of the water, they have to stay on the water, should not be submerged. In order to protect itself from the submerging, these bacteria have developed some special adaptation called gas vacuole. They have a bubble. That bubble which will have a lot of air inside. That air will make this cell to make buoyancy or floating. Help in buoyancy. Help in buoyancy or floating. Like that, the gas vacuoles are very much essential for the bacteria in order to maintain constant exposure to the sunlight to conduct the photosynthesis. Very important. Gas vacuoles are exclusive for only those bacteria which can able to perform photosynthesis. Could you name some examples of photosynthetic bacteria? Well-known one, give me. Very good. Cyanobacteria. Cyanobacteria. Like this, we have variety of structures inside a prokaryotic cell. Next, in addition to them, we should not forget something in the name of ribosomes. You know ribosomes? What type of ribosomes could you see in a prokaryotic cell? They are called 70s. Okay, done. I'm discussing about 70s ribosomes. Chemically, ribosomes made up of rRNA plus proteins. Ribosomes made up of rRNA plus proteins. Nothing else they have. This one you have already seen in your molecular basis of inheritance during translation process. Ribosomes made up of rRNA and also 80 different types of proteins. 80, the number also mentioned there. That's for 70 years only they have indicated. Next one. Because they have rRNA and proteins, all together they are called ribonucleoproteins. Ribosomes are known as ribonucleoproteins because rRNA and proteins are present in them. Next one. If you talk about the ribosomes, so-called 70S ribosomes, S stands for Swedberg unit. You might know that the Swedberg unit is a unit for sedimentation coefficient. Now, the 70S ribosomes, if you take, they have two subunits. A larger subunit known as 50S, the smaller subunit known as 30S. Remember, this larger subunit, smaller subunit, in a non-working or inactive state, they present as separate bodies. There is a ribosome floating in the cytoplasm. Catch it, allow it for sedimentation coefficient. If it has given 30s sedimentation coefficient, then this is smaller, 50s some other will give. Now, Whenever these two are joined, careful, during working in the sense, during translation, larger subunit and smaller subunit will join. Before and after translation, they will be present separately. By chance, you have got a ribosomal subunit which is participating in translation. At the time, if you sediment them, you will get 70s. That's the reason. Many are confusing. This is 30, that is 50, joining 80, but you have written 70. Now, 70 is sedimentation coefficient. Okay, it's not about subunit combination. When they join, and I will use it for sedimentation process, allow it for settle down, then it will settle down at the 70th area. If I individually allow it, it will settle down at 30th area. This one will be settled down at 50th area, but both are combined if join and then settle down, I will give it 70th area. Both of them combinedly. Diameter or width of the larger and smaller subunit together, it is around 15 nanometer. Length would be around 20 nanometer. Why are you mentioning like that? 15 by 20. Why? The reason is very interesting. These are the cell organelles which are smallest in the cell. If you find out any kind of cell organelle that would be very minute and smallest, you can see that units, they are nanometers, not even micrometer. They are in nanometer range. That's why they wanted to show these are the smallest cell organelles. Smallest cell organelles inside the cell. Out of all the cell organelles, whatever you will come across, 70s ribosomes are the smallest cell organelles in the field. Okay. Then another important thing about them, whenever they work, I mean ribosomes work is nothing but what? That is protein synthesis. Whenever they are doing protein synthesis, they have an interesting behavior like this. They will find one mRNA, in prokaryotes I am talking about, one mRNA. On one mRNA, several 70s ribosomes will join. Several 70s ribosomes will join. Several. Many 70s ribosomes would get attached to a single mRNA. Many ribosomes on single mRNA, this condition is known as polysome or polyribosomes. Now, this polyribosome generally developed by prokaryotic ribosomes or 70s ribosome. In fact, to say, wherever you could see there is a presence of 70s ribosomes, in all those places there is a chance of development of polysome or polyribosome. Wherever, I mean to say, this 70s ribosomes you will find in the matrix of mitochondria and also in the stroma of the chloroplast, and in different places wherever you identify the 70s ribosome, they do this work. On one mRNA, several 70s ribosomes will attach and they involved in different levels of translation events. Such kind of arrangement of many ribosomes on single mRNA condition is known as polysome or polyribosome. Next one. In a prokaryotic cell where these ribosomes are found, this is also another interesting thing, very, very important. When you talk about, okay, 70s ribosomes, they are attached to plasma membrane of prokaryotic cell. They are attached to Plasma membrane of prokaryotic cell. Observe very closely. Why you are specially talking about this? When we talk about eukaryotic ribosome, so-called 80S ribosomes, where do you think that 80S ribosomes could be seen commonly? On the top of endoplasmic reticulum, in the name of rough endoplasmic reticulum. That is 80S ribosome. But there will be a presence of prokaryotic ribosome, so-called 70S, would be attached directly to the plasma membrane. It doesn't mean that they will be present only on the plasma membrane. Some freely present on cytoplasm. What does it mean by this? Somewhere else we have seen one question in previous papers. There is a presence of ribosomes on the mesosome. That's correct. Mesosomes also can carry ribosomes. There is no exception for that. Okay. So 70S ribosomes either freely present in the cytoplasm, or they may be present in a status where they directly go and attaches to plasma membrane. Okay. Up to this, whatever the discussion I have made, this one is belongs to prokaryotic cell. Next discussion we will begin with eukaryotic cell. The next class we will begin. If you have any doubts, come on, you can paste it there. I can respond for that. Come on. Any kind of doubts. 22 question from exam paper, somebody says. What is that? Which paper you are talking about? Okay. I don't have that question. In fact, in the space exam syllabus, they given from the dicot route to end of the chapter, means tissue part is not there in the syllabus. No, tissues, say dicot route. If you begin from the dicot route, that means within the dicot route, whatever the tissues will be there, they will be noticed. Not in the tissue system. Tissue system part is not there. Okay, we'll meet in the next class. Thank you, bye.
Substitution for Indonesia.